Hello everyone, thank you for your presence. It's my first time in the region and I'm not an Anglophone, I'm a Francophone, so bear with me with my English. Maybe sometime it can be difficult, but I will try to do it as fluently as possible, especially it's recorded. So today I'm very pleased to present you some of our recent work. We've been working on this for the last year. It's not my main research topic. I'm mainly doing either pain pharmacology, very classical pharmacology, or I'm doing neuropeptide analysis and neuroprotein analysis. So that's my field of expertise and mainly a lot of proteomic work. So from that proteomic knowledge that we have developed and specifically targeted proteomics, I've been involved with Thermo a couple of years now down the road, and they liked the strategy we developed, so they wanted us to maybe apply that strategy to this problem of meat authenticity. We're very much into targeted proteomics instead of doing shotgun proteomics, and I will try to go with you on the workflow and try to convince you that the workflow we propose, it's a lot more compatible with routine analysis compared to what has been done in the past. I'm not sure what are your experience in proteomics, but generally in proteomics, we're very prone to identify a large amount of proteins, but there are very few explanation on the real role of those proteins into the biological system. So that's a challenge today in proteomics. We're very good at identifying proteins, but not very good at explaining the biology. So targeted proteomics avoid all this technical aspect, and we can really focus on what is really needed to understand within our sample. So today what I'm going to talk about is I will talk about the current methods. I'm not going to spend too much time because we had a nice presentation just previously. I'm going to speak about bottom-up proteomics workflow, a little bit about sample preparation, and in the afternoon Osama will present you the workflow, the preparation, the very simple preparation workflow we have for this method in the lab. Some data analysis and specifically the new strategy we propose that merge in fact bioinformatics with targeted bottom-up proteomic approach instead of untargeted approach. And I will present you some conclusion and perspective as well. So without going into much detail, we knew there was a big scandal in Europe with the substitution of beef by horse meat. And also they detect pork DNA in those mixtures, not only horse, so that was a concern. From that, we were all aware that for economical reasons, those companies are ready to do almost anything to reduce the cost of their product and sell it. So that was a problem, and the media has done a lot of stories around it, as well as the government, since government they were alerted but not keen to develop new laws. Now with those scandals, they develop new law and new criteria for product to be sold in supermarkets. So the motivation is dollars. So horse meat is a lot less expensive compared to beef. So they add horse to reduce the overall cost. And same thing with pork. Pork meat, generally, it's less expensive than beef, so they mix it with the overall product just to reduce the cost. So it's the main motivation for the company is cost. But at the end of the day, for us, well, it's an economic fraud. Obviously, you still pay the same amount, even though they mix it with horse or pork. It represents a health issue for certain person because some people have specific dietary restrictions that it may represent a challenge for them. It's an ethical issue, and it can be also a cultural and religious issue specifically in this area. So that's something we need to consider and respect as well. So traditionally, there was method using gel electrophoresis and Western blotting, qualitative real-time PCR or RT-PCR as well, even ELISA methods were used to do speciation. These methods are mostly qualitative, maybe with the exception of with RT-PCR. Molecular information obtained is very limited or almost none. The data cannot be revisited post-acquisition for data mining, so you don't really have data, you just have a signal that you interpret, so that's limiting. They are not generic approach and need to be heavily customized. So every time you had a species, you need to go through all the validation process, which require a lot of work before you can use it routinely. So these are kind of limitation in my opinions. So what do we know? What we eat, the meat are from animals, and we know that all forms of life are relative by common ancestry and descent. The constitution of phylogeny provide an explanation of the diversity seen in the world. So the diversity is based on DNA. So from that, today, all the phylogeny is based on the DNA sequence. So by having this information, why not make the relation between the gene and the species as central for meat speciation? With the RT-PCR, that's exactly what we do, but for our mindset in developing a proteomic-based assay, this is very important because historically, proteomics were working in a reverse way. We were looking at proteins, and at the end, we're upping the ladder and looking at the genes. But let's think reversely. So conceptually, we have DNA information from sequencing of different species that translate to messenger RNA that comes to proteins. And from these proteins, we can generate proteotypic peptides. And these proteotypic peptides can be fragmented with the mass spectrometer to form very specific fragment ions with the MS spectra. So there are two very distinct approaches. There are the shotgun proteomic approach, where we record a series of mass and we trigger MS recording based on either a mass list or it's done automatically by selecting a window of mass and generating MS spectra. Today, mainly, I'm not going to concentrate on this approach. I will concentrate rather onto a targeted MS approach. So we're selecting within a very complex full scan MS spectra, we will select an ion, will generate MS spectra, and will select very specific fragment ions to generate precursor to fragment chromatograms. So what's the strategy we start with? We can have the genome information, the transcriptome, the proteomes, so it makes the complete set of proteins that we could potentially observe. So historically, the experiment where we were doing a survey scan and survey all the precursor ions in a sample, acquire MS spectra for product ions, and we were surveying database like Mascot or Sequest using either Proteome Discover or other software that were available. So by doing that, we had massive data that we were comparing with a database that contained all the proteins. And we were using statistics to identify either, well, initially identify a protein or identify a species. And basically it was done by statistics. And I like to compare that as a fishing expedition, because depending on the quality of the data you have, the ion intensity, the number of hits you will have is variable. And it's highly variable by the user, by the technology you're using, by the quality of the sample, and in my opinion, it's not really compatible with a routine analysis of a food product or any product that you will try to perform routinely and try to have a quality control of that product. So what we do propose is we want to generate a proteotypic species-specific mass list up front, generate MS product ion species-specific mass list up front using bioinformatics. And after that, we generate MS and MS scans. And we survey this based on those lists to perform speciation based on specific peptide biomarkers instead of going toward a database survey. So we're doing this work up front instead of doing it afterwards. So that's the basic of what we will propose today. So very briefly, and you will see it later on, the sample preparation is very simple. So we mix the meat sample with water at a ratio of 1:5. So it can be 20 grams of meat with 100 ml of water, or it can be as little as 2 grams of meat with 10 ml of water. Even lower than that. We homogenize everything. We can sonicate the mixture as well. Protein is in the suspension are precipitated with acetone. So acetone precipitate the protein. In the supernatant, we have the mixture of water and acetone remaining. And the benefit of this is we're extracting fat that could interfere later on. So after that, what we do is we centrifuge, acetone is discarded, and we have pellets. That can be dry if we want. So it's not necessary, but you can dry it if you want to remove the excess of acetone. After that, we dissolve the protein pellets in ammonium bicarbonate at a pH of 8.5. And following that, the protein are denatured by heat. So we keep the heater at around 120 degrees, and we put the sample in there. We're not waiting that it's boiling, but near boiling. If it's near boiling, we remove the sample. So protein is denatured. Protein that are denatured are easier after that to perform digestion with trypsin. Okay, it may not be totally necessary, but if you want to have good efficiency, it's better to denature the protein. It's very classical. As an option as well, it's not mandatory, but as an option, we can reduce the cysteine bound with DTT and alkylate with iodoacetamide. So, but it's an option. It's not necessary. The data we generate this week, we haven't done this test. The data is very good. I hope it's going to convince you later on. And after that, what we do is we use proteomic grade trypsin, and we do a trypsin digestion. So trypsin will cleave proteins, in fact, at the C-terminal of lysine and arginine. So it's very predictive. And in fact, we use bioinformatics to predict which proteotypic peptides we will generate from the proteins. So the system is very simple. The concept is very simple. You can use an Orbitrap, a Q-exactive, and I recommend you use a Q-exactive, but you could potentially use a triple, quadruple, or even a linear ion trap. In my laboratory, we do have a linear ion trap as well as a Q-exactive. We test the method on both. It works with the linear ion trap. The only thing you have to do is to increase the in fact, the gradient time. Instead of being a 30-minute gradient time, it's going to be a 60 to 90-minute gradient to improve selectivity during the chromatography. But it's workable. So essentially, we will generate a survey MS scan, select specific ions, and acquire MS spectra. That's essentially what we will do. So obviously, when we start with this, we look at the literature. We saw very classical approach, so we said, okay, let's start with the classical approach and build some argument why we should not use this. So we collect data. In fact, this is an example of the total ion chromatograms of pork meat. We digest pork meat. And from the digestion, as you can see, it's very dense. And in fact, when you look at this, we have about 7,500 different peptides identified in there, okay, with a very high threshold. Most likely, if we reduce the threshold for identification, we will identify over 10,000 different peptides. So we spend a lot of time in surveying database with all these peaks. And in fact, when you do this with different species, we did it with beef, horse, pork, lamb, and even with chicken, we concentrate initially our effort on mammals because differentiating mammals with a proteomic approach is more a challenge than other species. So that's why we concentrated there. So what we, after dozens of experiments, we identify 7,429 common peptides. So they have the same sequence, same mass, same MS spectra. And we were able to identify 17 specific beef peptide, 14 for pork, 9 for lamb, 23 for horse. When we saw this, we said, okay, this is a pretty convincing argument that we cannot use this approach to truly do speciation for routine analysis because this data will be too variable to really validate at one point and get that going into a lab that has multiple samples. So we decide this is a very convincing argument. So just to show some historical data as well, this has been published following the scandal using exactly the same approach. And I hope this will also provide some convincing. So in fact, when you look at this, this is an example of the data they generate for pork, horse, and beef. Every single color is replicates. So it's been done in triplicates. And as you can see, for each species, they have different, not necessarily different peaks, but different abundance. Sometimes it's more abundance, sometimes it's less abundance, and significantly less abundance. We don't see it very well in there. I'm sorry about the quality in there. But there are a lot of peaks they use to identify a species that its own specified signal. How can you base speciation on unspecified signal? It makes no sense. I mean, but this was used. And in fact, conceptually, it's a bit like a fingerprint. They use it as a fingerprint. And we know that fingerprinting, it's not the best way to go. And especially in that field, you have a company that is doing, obviously, adulteration to reduce the cost. They are doing economic fraud. If you go in court with this type of data, it's not going to last long. A good lawyer will destroy that right away. So that's not the way to go, not in our opinion, though. So all of this was pretty convincing. Our initial data was compatible with this, and there was this preliminary paper. There are other papers later on that build on this and try to improve this, but essentially, they're still doing this. So, what can we bring to the table? So, what we try to do is not rely heavily on the quality of the MS and the DMS data. So it's already set. So people can just run their sample. They don't have to think too much about the setup itself. It will not rely on bioinformatics and parameterization. So bioinformatics will be done up front. And you're going to have a list of mass, and you just have to survey those mass. It will not require high-skilled scientists or highly trained people in proteomics to generate high quality of data, because that's not the reality. Especially in food chemistry laboratory, people are not trained in proteomics, so it's not realistic. So yes, we can propose an alternative to this. So let's start again with the same concept. We have the messenger RNA, so we can predict proteins and we can predict proteotypic peptides. So again, from that, we can generate very specific mass lists derived from proteotypic peptides that will be specific to one species to the others. And we can also generate a very specific MS or product ion database based on this knowledge, because all this can be generated in silico. So we can, at the end, generate post-analysis extracted ion chromatogram for selected proteotypic peptides and perform speciation based on this. So I'm going to use this example. I'm going to use myoglobin as an example. In fact, when we do try to quantify subspecies or adulteration, we use myoglobin. Myoglobin is a very known protein and is highly expressed in muscle tissue, so it's very easy to extract, very easy to analyze. But there are others, and I will show you others. So when we perform our analysis, so we extract gene information, we perform in silico sequence alignment. After that, we perform in silico protein translation and alignment. So we align the proteins so we can compare from species to species. And we perform in-depth sequence analysis. So when we compare this, we can identify area of the protein that are distinct from one species to the other. The advantage of this way of doing the work is if I had other species, it's a lot easier because I know exactly where to look. I know where to look. So we can generate this way a very specific mass list. So when I align them, okay, and I select the potential proteotypic peptides, I can generate the mass because the mass is associated with the specific sequence. And I see the sequence are not common. There are differences. From that as well, when I look at specifically myoglobin, I can identify other specific peptides, especially for pork. So I can still use myoglobin and monitor other peptides that are very specific to the pork. So not only I can use our specific proteotypic peptide, but I can use others. I'm not going to show the data for this, but it's available. And that's the beauty as well of using a Q-Exactive in full scan, because I can mine the data and extract those information afterwards. So that's a very important thing. So there are others. So we did the same thing with myosin 1 and 2. So we can identify an area of the protein that is highly distinct. There are almost 2,000 amino acids on myosin. If you look at it, this is the only area I can truly distinguish them. So if I do that up front, I can target it. But if I don't, I'm just fishing peptides along all those sequences. And the question is, would I be able to detect this one with the amount of data? That's the question. So by knowing it in advance, I can fish for it right away. So same thing with beta hemoglobin. We identify two areas, mainly this one, but we can also survey this area for beta hemoglobin. So the other question is, when I look at those sequences and the differences between the sequences, and you align them, and I look specifically, okay, into the MS2 fragments, what I see in there, and it's very important, there are distinct fragments for each of the species. So not only the precursor ion are distinct, but I do have distinct fragment as well. So the pair of precursor to fragment ion are very specific to each species. So the chance of false positive or even false negative is almost none. So this is a high strength. So same thing with myosin, myosin 2 and beta hemoglobin. So When we look at it more in detail, peptides are always fragmenting with the same patterns, mainly with the type of instrument we are using, with a low to medium energy collision cell. We get Y and B fragments, mainly. Sometimes we do see A and X fragment, as well as C and Z fragment, but generally they are the least abundant in the spectra. So our objective is to be able to detect the peptides, so we're not concentrating on them, we're concentrating on the most abundant one. So generally it's B and Y fragments. So when I look at the statistical analysis we have done previously using the Venn diagram, we can isolate which fragments are very specific. And when we look at myoglobin with pork, we can find 3 specific fragments. These are, in fact, you will learn it later on, Y13 and Y14 fragments, and these are the fragments we will use, in fact, for this survey. And with ores, we use the same Y13 and Y14 fragment, and as you can observe, they have different masses, but they are the same fragments. So this is very interesting. And for myosin, 1, 2, and beta hemoglobin, there's a list of fragments. And where you see the asterisks, in fact, are the most abundant fragments we do observe in MSMS. So you could use them, potentially, to identify the species. So either to do authenticity or to do adulterations, you will be able to use those fragments as well. So when we look at this, I have my myoglobin proteotypic peptides, I have the different list, and I have this mass list. So I'm doing a survey scan at very high resolution, at 140,000 full width half mass. We're surveying between 500 to 2,000 in full scan. And what we will do is initially survey those mass. And following that, we'll acquire MS-MS spectra with an isolation window of 1.5 AMU, and these spectra will be acquired at 17,500. So, and we will generate post-acquisition XICs. So everything, all the extracted ion chromatogram are the mass, either the precursor or the fragment mass, with a 5 ppm window. Okay, that's very common in the high resolution world. So when we look at this, so here we have the total ion current for beef, horse, pork, and lamb. And as you can see, it's very dense. It's the same I showed you before that we could extract minimally 7,500 distinct peptides. So from that, when I extract those theoretical mass, I get a very specific peak that I can extract from that. Very, very specific for each of them. And this is very revealing. So I'm from this where there's a lot of peptides and I'm able to get this. So that is very highly revealing. So each targeted peptide can be detected and extracted from the TIC, from this complex TIC. So when we look at the MS-MS spectra that we collect, we can see the region that is very distinct, the Y13 and the Y14. And this is obviously compatible with the in silico analysis of the sequence we have done previously. So we can truly see Y13 and Y14 ions in each of the species. So we can distinguish them. So from that, we will select specific ions, precursor ions, generate the MS-MS spectra, and we'll extract those fragments ion that are specific to each species. So when we do this, and that's exactly what we have done there, here it's a 10-minute window, before that was 47, that's why the peaks don't look exactly the same. But you get the Y13 and the Y14. The other colors at the bottom are the other species. In this case, this is beef, and you see the large peak for beef, but all the other species, when I extract the same masses, I got nothing. It's very, very specific. So it's the same with pork. I can see there's no interferences from other species. Very, very specific. And I can see this with Y13, Y14 for each of the species we test. And in fact, we test also chicken. Later on, you will see chicken data, same thing. And obviously, it's easier with chicken than all these mammals. It's for genetic reasons. So is it quantitative? Yes, it is. Somehow it is. For sure, people that have background with small molecules don't expect the same type of quantitation, but for this type of quantitation, it's very revealing because we are able to have kind of a linear relationship between the percentage of pork meat in beef compared to the intensity of the peak we observe. So it's very linear, very reproducible. When we overlay the spectra, they look very nice. The signal, well, at 1% it appears low, but it's the same scale, so you can still see it, and we can go down. In this case, we went to 1% because of the regulation, but we could go lower than that. So when we look at the variability, these are 4 samples from different supermarkets. I went and buy meat sample from several supermarkets at different areas. They were not even in the same areas, so they could not be the same suppliers. I don't think they were the same suppliers, and they were done in triplicates. So in general, we get something within 20% coefficient of variability in terms of absolute signals. There is no internal standard, there's nothing simple. Preparation is very simple. It's quite good. I mean, it's very surprising. We were extremely surprised, okay, to see this. That wasn't our expectation because that's not our experience with proteomics in general. But using myoglobin, that's exactly what we get. After that, we did look at the optimization of the trypsin digestion. What you see is a very typical enzymatic reaction profile, a kinetic profile. So what you get is there's a starting point where the slope here is quite high, and at one point it does saturate it and it stops. In fact, the reaction is most likely saturated at all time because you don't have enough trypsin for the entire proteins that are present in the sample. But when we monitor the appearance of the product, that's the rate of the appearance here, and slowly it's shutting down. So after 8 to 10 hours, it's pretty stable. So an overnight digestion do a good job. Generally in proteomics, the people are leaving the sample for 24 hours and they just use it after. But after 8 hours, you can use the sample. Even we tested, well, this is the data, but we have other data as well. We tested after 2 hours or 4 hours, and you can detect the peptide. So if you need a rapid answer, you can take out the sample and analyze it, take an analytical, and leave the rest of the sample into the incubator. So the following day you can re-inject, you're going to have more sensitivity, and you will be able to confirm what you have seen as a first analysis. So this was very interesting. So just going briefly, all the other proteotypic peptides we have identified, we have seen them, and the delta mass is within 2 ppm, even within 1 ppm all without a log mass. It's very easy to find, and they are abundant, so you will find them. There's no problem. So when we look at spike meat at 1%, more in detail, you look at the myoglobin peptides and myosin 2 and hemoglobin, and you see a very large peak, very specific. And in red, what you have is the species, like this one, hold up. Here it's beef, so here it's myoglobin, and in red it's only beef, there's no pork. In the peak here it's the peak pork for myoglobin. Here it's the myosin 1 peak at 1%. Same thing, the myosin 2 peak at 1%, very clean. And this is only in full scan MS at 140,000 full width half mass. So it's at high resolution. Most likely you're going to get even a better results with the QE plus at 280,000. So if you can afford it, it's the way to go, I believe. So in full scan MS, yes, you can do it. And you have plenty of sensitivity. Even, I will say, and it's related to your question early on, in some case, it's better to work in full scan MS if you can, especially at high resolution, because when you do fragment peptides or small molecules, in fact, your ion abundance is distributed with the number of fragments you generate. So more fragments you have, less ion abundance for each of the fragments you have. So sometimes it's a trade-off you have to do. So it can provide you more selectivity, but maybe less total ion abundance. So sometime in full scan MS will pay it off for detections. So there is another very interesting advantage of this is when we know exactly which peptide we are targeting, we can do real quality control. And how we can do real quality control is we can synthesize cold peptide, so it's the real peptides, and we can synthesize heavy labeled peptides. So we can label the peptide with C13, nitrogen 15, even deuterium. I'm using deuterium quite often. It works well with small molecules. Sometimes it doesn't work that well, but for peptide it does work well. So deuterium is a cost-effective way of doing it as well. So if you're cost limited, that's something you could do. So this is very interesting because either you can use it as an internal standard or you can truly use it as a quality control measure. So you can add it to your sample and follow the doublet. So all the time. So you have both spectra from your internal standard and the peptide you target in your sample. So you always have, in terms of mass, you always have a doublet of mass that you can follow. So strategically, you need to put your label to follow this doublet of mass. So it's a double confirmation measure. So that is very good. And it's a true quantitative measure. So this is very interesting. So just briefly, we also tested with data-independent acquisition methods, which I'm not sure if you're familiar with, but traditionally in proteomics, we're doing this because we want to collect as much data as possible and after that, interrogate database. But it's very informative as well if we want to do data mining. So if we are doing some research work or survey work, we're not sure, we want to detect things that we are not sure what's present in the sample, that's one way of doing it. So what we do is we're having a wide isolation window. In this case, with the instrument I use 50 Dalton. You can use 25. There are even methods developed with 10. So depending of the instrument you have, if you have a fusion, you can work with 10. If you have a QE, you work with 50. Sometimes you can work with 25. With a QE plus, you could work with 25. Essentially, what we did, we wanted to compare MS1 data and even targeted MS2 data with DIA data. So our peptides has a mass of 744. So we extract the MS-MS data from this window that we're collecting MS-MS spectra from mass between 700 to 750 and extracted very specific fragment ions. And when we look at the linearity, it was pretty convincing as well. For sure, we're not collecting the data at the same rate, so we see differences, but it's still pretty convincing. This is very linear, but this is very linear as well, and we could detect it. So we were very, very happy with this. So when we look at this in detail, because we want to see the chromatograms, we want to prove that it's really there. So when we look at the beef samples that has been spiked with 1% pork meat, we can see here the nice myoglobin peaks, very nice at 1%, and you can see it's clean with beef that is not fortified. And when we look at the targeted MS2 data, what we can see, and we overlay both spectra from extracted ion chromatogram from both fragments, and you can see, first of all, the ratio that respect the MS spectra, but also very, very nice and sharp peaks in, we don't see it very well, but downward you can see pure beef that it's in blue and there's absolutely no trace, no interferences. And same thing with the DIA, we could see it, but there is much less data points, so it looks like it's smoothed out, but in reality it's because there is less data point. But it's still interesting that we could detect it using a DIA approach. It's shown the specificity of the fragment ions. So, and this one is a mistake. It's not supposed to be pork, it's horse meat. So we did the same experiment with horse meat. And we show, and horse meat were fortified at 1% with in beef meat. So we show essentially the same thing where we can detect the myoglobin peptide very easily. And following that, we can detect the fragment ions that are very specific, and it's very, very specific. And when we look at the appropriate windows, we also extracted the very specific fragment ions, and again, very, very specific. So I hope I convince you we can detect adulteration on beef meat sample with pork meat and horse meat as well, okay? So the general strategy, and this is what we wanted to propose up front that is very distinguished from what has been done in the past, is we really want to start with the genetic information and perform sequence alignment in silico, perform protein translation in alignment, analyze the protein sequence, and generate in silico tryptic digestion mass lists. So from this information, we can generate mass lists that are very specific to one species to the other, and generate MS product ion mass lists that can be used to select very specific ions into a very complex data set. And from that, generate post-analysis XICs that will allow us to perform speciation and detect adulteration. Right now, method can go down to 1% very easily. We've done it in the lab here without a lot of time, a lot of training. We did it once, it works, and we're very happy. In our lab, we went down to 0.2, 0.1%. But we think we can scale down this very easily because we have protein. The quantity of protein is not limited, so we can scale down this very easily. So we can have a very sensitive methods using this strategy. That's what we think. So I hope it convinces you. So I want to thank one of my students that worked on this. It was a side project with him. It wasn't his main project. So he helped me to generate all this data. And Alberto right now, he finished his master's degree and he's going to start his PhD very soon in September. And these are other team members that contribute indirectly to this project because they're working in proteomics in my lab. So they're developing different tools, different strategy, and I just use them and I'm just here in front of you. But really, these guys are really good and they helped me to be able to make this presentation. I want to thank them especially. They're really dedicated people. And I want to thank Thermo for this opportunity. It's a tremendous opportunity to be here and to work on this project. Opens a lot of different opportunity for me, for my team, and I'm very grateful for that. And I'm funded, it's a Canadian research funding, and I'm funded by INSERC, so I'm very grateful for them to provide funding for this. Thank you.